So welcome to the HPL seminar series. I guess since this is the first one this year, I'll introduce myself. I'm Olivia Bruce. I'm a PhD candidate working with Dr. Chapter. This year we will also have a co-moderator, Michael Bagley, who is not here this week. So today we'll be hearing from ScottSibole. Scott earned his bachelor's in biomedical engineering from the University of Akron, and then worked at the Cleveland Clinic Learner Research Institute as a research engineer. He joined the HPL in 2013, completing his master's work with Dr. Walter Herzog and Dr. Cellular. And that was on image processing and segmentation of chondrocytes. He then continued in the same lab for his PhD, and he recently defended in July. So Scott's research interests include continuum mechanics of soft tissues, image processing, software engineering, and modeling energy usage in buildings.
Switching it up.
So looking forward to sort of following these interests, moving more towards the now, outside of research, Scott mentioned that he's been rekindling his interest in chess. So,
very cool.
So today, Scott will be presenting towards understanding multiscale articular cartilage mechanics.
Thanks, Olivia. So jumping in and explaining the title, what do we mean by multiscale specifically? We have this idea, well, this problem basically in biomechanics, where we have a huge disparity in scale between the things that load the tissues and ultimately the cells in our body and our daily activities. So walking, running, jumping, climbing stairs, etc. We look at that as biomechanists, right? That's at the scale of about a meter. That, of course, gets distributed to the joints in our musculoskeletal system and to the tissues that compose those joints, and then further down all the way to the cells that are within those tissues. And if you look at that from a breadth of scale, you're going from the meter down to about tens of micrometers. And 5 orders of magnitude is quite difficult to relate things like forces, strains between daily activities and ultimately the biology that's happening at the cellular scale that we care about, because we know that the cells are very much influenced by the mechanics they feel for their activity. So we look at this experimentally and with modeling, and attempts have been made to scale all the way from the body to the cell. This talk is predominantly going to be focused on the interface between the tissue and cellular scales, so going from that centimeter scale down to that micrometer scale. And then the other half of the title, the tissue that we're looking at, is articular cartilage, which here's a human femur that has some exposed cartilage. The shiny tissue on the ends of the femur there is articular cartilage. It's very thin, 1 to 6 millimeters thick in humans. It's importantly avascular and aneural. In particular, the lack of a blood supply limits the cell metabolism. So in turn, the cells have very limited ability to repair the tissue. Mechanically, it serves mainly to produce a low friction sliding surface between the two bones that are articulating, and also to distribute loads over a larger area and thus reduce their magnitude. If we zoom in a little further and look at a histological cross-section of cartilage, we see 3 distinct classical zones of differing properties through depth, the superficial, middle, and deep zones. And in those zones, things like molecular content vary, the orientation of reinforcing structures, particularly collagen, and also the cell, the shape and distribution of the cells within the cartilage. And then if we go further below the deep zone, we transition from articular cartilage into bone and what's called the calcified cartilage region. And then below that is subchondral bone, which is very similar to cortical bone. And then zooming in a little further, if we look at a transmission electron micrograph of a single chondrocyte cross-section, we can look at very closely the interface between the cell and its surrounding environment, which we call the matrix. And one of the things we can see in this picture, if we outline and highlight, is that the membrane of the cell is not smooth. It has these ruffle features on it. and these ruffles are actually distributed in a non-uniform way around the circumference of the cell. And then the other thing I've highlighted in the image in yellow is it's a little hard to see on a TEM image like this, but a distinct specialized region called the paracellular matrix that directly surrounds the cells in your cartilage that differs in molecular composition, mechanical behavior, and so on from the bulk volume of the tissue, which we refer to as the extracellular matrix. Looking at cartilage from a mechanical engineering standpoint and looking at its material properties, we can say that it's both poroelastic and viscoelastic. These two things give rise to time rate and history dependent properties, namely stress relaxation and creep, which I show on the right. And we model these as, in poroelasticity, we model it with something called biphasic mixture theory. So basically we consider the material as a mixture of a solid and a fluid, or even more complicated, might consider a solid, fluid, and ion phases, and actually consider the movement of charged molecules through the tissue. In addition to this complexity, it's also heterogeneous, as I talked about earlier, in particular with depth, and anisotropic, which means that it behaves differently depending on direction that you load the tissue. And this anisotropy also changes with the depth in the tissue. So I showed you these different scales of articular cartilage. How do we go about measuring or relating mechanics that occur on, say, the tissue scale down to the mechanics that the cell might be feeling? And one of the approaches that's been popular for the last 3 decades or so is using what are called optico-mechanical systems, where we couple a device that can image the cellular geometries, in particular a laser scanning microscope, which can obtain 3D images of microscopic volumes of cartilage and the cells within it. And we couple those to a mechanical testing system where we can apply some controlled known tissue scale mechanics. In our particular lab in the basement, we have a setup where we can indent the cartilage, basically poke it, or we can have another setup where we can apply what's called confined compression, basically a 1D sort of, we don't allow the tissue to expand, but we load it in a single direction. When we apply those loads, we can simultaneously obtain 3D images of the cells within there. And so we have a picture of the cellular scale geometry synchronized with tissue scale loading. And so in that way, you're sort of doing a multi-scale experiment where you can observe the relationship in those mechanics. So I mentioned laser scanning microscopes can obtain 3D volumes of cells or of cartilage, and those look like this. So we have a particular volume of cartilage with cells there, as you see in the darker blue objects are cells, and then the same volume under some tissue scale deformation. And you see that it's changed, it's deformed. So going from something like this, which is just an image, a 3D image, to deformation, we need to somehow quantify the shape of those cells within the image. And that's a discipline called image segmentation, which is a very old problem. It's been challenging. Mathematicians, physicists, engineers have thrown basically everything but the kitchen sink at it. Thresholding, active contour models, topological analysis, machine learning methods. And once you apply these, and probably a collection of these, you'll hopefully arrive at a solution. And in the end, you get something that looks like this. We won't go into the details of all the math behind that, but you get these triangulated surface representations of the cells. And these are what you see in like a Pixar movie or a video game, same idea. We have a very computational numerical representation of the cell shape from these. So now that you have a numerical quantification of shape, how do you calculate deformation between, say, those two unloaded and loaded states? So historically, the first thing that was done is from those triangulated surfaces, you can directly calculate the volume and surface area. So you can look at the change between unloaded and loaded states of the cell's volume and surface area. The present state of the art prior to this work was to approximate the cell shape as an ellipsoid through various techniques. The most popular of these is looking at the principal moments of inertia tensor, are the principal moments of the inertia tensor, and an equivalent ellipsoid that reflects that inertial tensor. And essentially you get ellipsoidal lengths, and you can look at the change in the lengths of those ellipsoid axes and calculate sort of normal strains from those. We chose to look at it in a little way, a more classical continuum mechanics idea of how deformation is defined. So if we consider an arbitrary body and some unloaded reference configuration on the left, this label as reference, and all the points on the body we call big X, say, we can imagine that there's a mathematical function that for every big X uniquely maps to a deformed coordinate in a deformed body called little x. And this function, we would like it to be 1-to-1. So for every big X, there's only one little x. And also we want to be able to reverse this process, so it needs to be invertible. And so, as you said, we can go from little x to big X, or from a big X to little x. You can imagine a general function that could describe the deformation of an object is not really tractable to find in the real world. So instead, what we tend to look at is we attach little local coordinate systems to points of interest on the body. And in that coordinate system, we consider vectors that emanate from those. In the reference configuration, we call those tangent vectors, say, big M. We use big letters for the undeformed configuration. And then those get mapped to some deformed vectors, little m. And we assume that mapping is a linear equation, which is represented by this is essentially three equations in the bottom. Little m equals this boldface f times big M. So for every vector, we can imagine this linear function, set of three functions that describe its deformation. So very close to this linear transformation is something called an affine transformation. And you're all familiar with an affine transformation in 1D. That's simply the equation y equals mx plus b. So mx is a linear equation plus b with this translation that makes it affine. In 3D, it's that equation that I showed you before, plus a translation vector x naught. And so basically, f has 9 unknown components and x naught has 3 unknown components. So we have 12 unknown components. We can do some sort of inverse, we're solving an inverse problem there, looking for those components of f and x naught. We can do that with an optimization technique that looks something like this. If on a cell we attach at the centroid in the reference configuration a coordinate system, and then likewise that same cell in the deformed shape of that cell, at its centroid we attach a coordinate system, align those coordinate systems and call that origin x naught. And then we consider all the vectors emanating from that to the surface of the cell. Those are those tangent vectors I talked about, big M. And then we simply optimize f and x naught applied to these red vectors to match the surface of the green object. And when we solve that optimization problem, we get what's called the deformation gradient, which is a very, very important quantity in continuum mechanics. Basically, we can describe any sort of strain measure, anything that you learn in your undergrad engineering courses is a function of f. Once you have f, you essentially have it all. And so for the particular cells I showed you, we can visualize that deformation gradient as going from the outline box in red to the green deformed box. So you see there's shears and compression strains occurring on that particular cell. So I implemented these in two open source packages, one for my master's work called PyCellAnalyst, and then a new software package I'll talk about later called Resonant LSM. On my GitHub profile, my username is ScottSibole. You can get any of these. They're licensed with open source licenses, and so free to use for whatever you want. So extending the previous state of the art, we go from ellipsoidal approximation of cell shape, where we can get an approximation for normal strains of the cell, to getting a full deformation, assuming uniform deformation across the cell, which is still a limitation, but it's a little bit further description of the deformation of the cell. Okay, so cellular deformation is important, but so is tissue deformation. So for that, past methods looking at the microscopic images, the simplest technique would be simply to look at the like a 1D strain, a change in distance between cell centroids. So if you imagine the cells highlighted or circled in white, the distance between those centroids to the cells outlined in yellow, the distance between those, you can get a change in length and get sort of a 1D strain measure from that. In my master's work, I extended this. If you consider the centroids of 4 cells, you can build a shape called a tetrahedron. And then from that tetrahedron, you can calculate the deformation gradient again, as I said. That's the minimum sort of shape that you need to calculate that if you compare the shape of tetrahedrons we formed reference and deformed configurations. The problem with that approach is that in cartilage, the distribution of the cells is quite sparse. So you don't have a lot of trackable features. So you're going to be assuming uniform strain across quite large areas. So we came up with the idea of using a feature of laser scanning microscopy where you can do what's called photo bleaching, which essentially you can draw on the tissue in a non-destructive manner, whatever you want. We choose to draw a 3D grid as I'm showing here on the left, reference undeformed cartilage volume. And then we apply the sort of same mechanical testing experiment and deform that tissue, image it. And what that does is by having a grid on there, we've increased the number of like trackable image features in the image. And we can do a mathematical technique called deformable image registration, where we approximate, if going back to the picture of the deformation, that function chi, we approximate that with, in this case, a third order basis spline. You can do it with whatever transform you want. This is what I chose. We minimize a sort of, in this case, correlation between pixels so in your reference image, you have a pixel, you apply your guess for what the map is, you compare it to the pixels surrounding it to the deformed image, and keep doing that until basically the correlation is maximized or minimize the negative correlation here. And what the grid does is it greatly increases the robustness of this technique. So in the end, you can get very accurate measures of deformation across the entire image microscopic volume. And from that transform that you calculate, once you get an optimal transform, you can get something like a displacement field. You could apply that to the volume of tissue that you looked at and visualize its deformation. So the reference is, again, the outlined wireframe box to some deformed configuration as the solid gridded region in the image, or in this slide. You can calculate any kinematic variables, third principal strain, which is like the peak maximum compressive strain, volumetric strain, visualize it in all sorts of ways. Do a lot of things with that. This is, again, implemented in an open source package called Ellison GridTrack, which is available on my GitHub for free. So extending tissue deformation analysis, we go from potentially measuring a deformation gradient against across large volumes of tissue to very fine and robust measurement of 3D kinematics across the whole imaged region. This work is published in Active Biomaterialia if you want to read about it in detail. Then moving on to a slightly different topic, when we were doing this grid tracking, this photo bleaching experiment with deformable image registration, we kind of accidentally found this new tissue stain that we used for the specimen preparation that would distinctively leave these regions of low intensity around the cells. As you can see in black here, I'll highlight around, say, a couple of those cells, which are the blue-red color map. And the idea occurred to us that we could maybe use this stain to measure the geometry of that paracellular matrix I talked about earlier. And the important thing about this is that the previous technique to measure this structure, this paracellular matrix, required tissue fixation was with immunostaining, which required tissue fixation, which kills all the cells in the tissue and also modifies the material properties of it. So the idea being, if we want to study the mechanics of the paracellular matrix in living tissue with unaltered material properties, maybe we could use this stain. So we did some experiments and wrote some software to do just that. The software could segment out the PCM. We found that was quite reliable. And we wrote some additional analysis software to measure how the thickness of the PCM changes circumferentially around the cell. Applied that to a statistical sample of around 350 some chondrocytes, and saw a very distinct property where the paracellular matrix had an asymmetrical thickness around the cell. So about twice as thick a paracellular matrix below the cell as above. And so this is very strong statistical observation. Does it have any meaning? So we, well, first of all, the segmentation and analysis that I just presented is available in a software package on my GitHub called PCM Segmenter. But the idea being, is this asymmetrical difference that's distinctively biased towards a thicker PCM below, is it important? Could it be important mechanically? I'm a mechanical engineer by background, so I think about mechanics. So to look into that, we did something called multiscale finite element modeling. So basically at the cellular scale, we made a model, a simplification of the cell geometry, the paracellular geometry that we observed on the far right, so 54% thicker on the bottom than on the top. A symmetric model where the top and bottom of the paracellular matrix were equal, and then the opposite of the observed reality, where it's 54% thicker on the top. And to drive these cellular scale models, we needed information from the tissue scale. So what are the mechanics of that cell are feeling? How are we going to deform that tiny microscopic volume containing the cell? We need that information from a tissue scale model. So we have a simplified model of full thickness cartilage where we capture the depth dependent properties through how they vary throughout the depth. Solve that model, strain to say 5% compression, and we take the result of that and essentially interpolate those to the outer perimeter of those cell scale models, and they serve as mechanical boundary conditions for that. And in the end, we get a sort of a simulation of cellular deformation from those, and then we can compare the different symmetry cases that we consider. And so again, on the far right is the real case, what we observed. Far left is the complete opposite of reality, and the middle is symmetric top and bottom. And the distinctive finding was that this cell, when you, in the fast onset, early period of loading, would experience very high fluid motion around it. And the cell being basically impermeable to water would be dragged up. So essentially rigidly translated vertically. And this resulted in a strain, a tensile strain in the paracellular matrix below the cell, which is, here I have my, so here, this region here, you see this bright red, and it's less bright on this because it's lower. So realistic case, we have lower tensile strains developing below the cell, highest tensile strains in the opposite of reality, and somewhere in between for the symmetric case. So what does this mean for the cell membrane? The paracellular matrix is experiencing these tensile strains. These are translated to the cell membrane as well. You get these highly localized, high, unnatural, perpendicular to the membrane strains occurring on the bottom part of the cell in this very early stage of loading. And then as time progresses and things slow down, the motion of the fluid slow down, you get what you more expect in a sort of thin membrane loading scenario where all of the strains are oriented tangential to the membrane. Okay, so that's the bottom panel. So what could those high strains on the bottom result in? Well, one, cellular detachment. So cells attached to their surrounding matrix by these protein complexes called focal adhesions. They spend energy to make those focal adhesions. And when they detach, those focal adhesions are essentially lost. They stick to the matrix, and then the cell would have to regenerate them. So energetically, it's bad for the cell, but probably more importantly is that you're going to drastically change the mechanics currently felt in the cytoskeleton of the cell, which then could have huge implications for its biological response to that. Worse than that, potentially, is this highly localized non-uniform strain might lead to membrane rupture, similar to imagine poking a soap bubble with your finger. Your finger introduces a very highly localized perpendicular strain to the membrane, and that can rupture the lipid membrane, just like a soap bubble. And then finally, another anecdotal observation, which we need to explore further, but going back to that electron micrograph image, remember those ruffles, they were distributed non-uniformly, but they're also concentrated on the bottom of the cell. So potentially this ruffled-like structure, and we have shown this in other experiments using atomic force microscopy, could act as a sort of strain reservoir to protect the cell membrane from rupture. And maybe in conjunction with this sort of behavior that we're seeing from the finite element model, this could work in conjunction with the thicker PCM below as a protective mechanism. The multiscale modeling project here is fully automated. You can generate other multiscale models with a software package called BioMultiscale, again, available for free on GitHub. And this work is published in Annals of Biomedical Engineering if you want more details. And that leads to our final project, which has actually been the long goal of our lab. We acquired a microscope about 6 years ago, I'd say, that's able to measure much faster than other laser scanning microscopes. The idea being we want to measure faster rate mechanics of the cells, what I'm calling transient cellular mechanics. Up until this point, everything has been essentially a static loading condition. We waited till things settled down. We're at an equilibrium state. We have plenty of time to acquire a 3D image of the cell and analyze it. But we'd very much like to acquire things on the order of seconds, where we can look at what the cell is experiencing in response to tissue level loads that might be reflective of something like gait, say. So the limitation of the microscope is mainly due to what's called the rasterization process. Essentially, when we image a particular plane, a focal plane, we have to go row by row, just like your TV or your computer monitor does, and draw a line row by row raster pattern across the image, moving the laser. And to move that laser, you have to adjust mirrors, which are controlled by these little servo motors. And a lot of work has been made to make these servo motors move really quickly. So in a traditional scanning microscope, you might scan out 4 rows of that focal plane in about 20 milliseconds in this little sawtooth pattern I'm showing on the right. Alternatively to using the servo-controlled motors, you can instead resonate the mirrors. So imagine a tuning fork, you hit it and it resonates at a frequency, right? You can do the same thing with the mirrors, just resonate them. And rather than a sawtooth pattern, you get the mirror changing position like a sine wave on the bottom. The advantage of this is being that you can scan out those 4 rows in about 200 microseconds, so about 100 times faster. Drawback is you get lower image quality for a number of reasons, which I won't go into. But the question was, can we analyze these lower quality images and still get the meaningful information that we're after? And so we wanted to look into this. We chose a fairly tame dynamic loading condition where the cartilage is experiencing what's called osmotic swelling. So basically we put the tissue in deionized water. Water is going to flow into the tissue, cause it to swell up. And we want to image microscopic regions within the tissue at different time periods throughout that swelling process. And then of course, develop software to analyze the data that we acquire there. End of the day, we were able to do this quite well. This is what the images look like with resonance scanning. The cells that we segmented out qualitatively, it looks quite good. We of course want to have quantitative validation methods though. So in addition to these qualitative measures, I also developed a validation testing framework where I could generate synthetic images reflective of the image quality of the resonance scanned images at varying degrees of image quality. And then we know exactly what the geometry of those cells are. We know exactly what the deformation of those cells are. We can compare that truth, that ground truth, to our segmented results. And what we found was very, very good agreement. Median errors on near 0%. The range of errors less than 5% in, say, volume and strain, for example. Also, in addition to the cellular deformation, we're interested in the tissue deformation occurring at those faster rates. And for that, we wanted to extend the deformable image registration technique where we use the photo bleached grid. Now our resonance images look like this. They're much grainier, a lot more speckled noise and stuff in them. But in the end, we found that it didn't affect it too badly. We got registration errors on the order of displacement errors on the order of 1 micron, which is essentially the resolution of the image. And then a final point, we need to explore this further, but we did this osmotic swelling experiment simply really as a proof of method, just a demonstration experiment. But there was some meaningful scientific findings in that what we observed was the cells, when exposed to this sort of swelling condition, they swell up initially, as we expect. But given enough time, rather than continuing to swell or maintaining that swelled volume, they start to return to their reference volume, which indicates that there's some process using energy to regulate the volume of the cells. And so this needs to be explored further, but we know that the cells can actively regulate things like their volume and activate their and regulate, say, their cytoskeleton and their mechanics through spending ATP to do that. But previously we couldn't observe that on this time scale. So this is something that we found quite interesting and we should explore more. And finally, again, you can get all this in the software packages on my GitHub. And this work is published in a journal Biomechanical Engineering. So summarizing it all, the work mainly focused on developing new tools and methods to look at the relationship between tissue scale mechanics and cellular scale mechanics in articular cartilage. We get tissue strain now from deformable image registration. We have a method to image the paracellular matrix geometry in tissue containing living cells. There's a software package to do automated multiscale modeling at the cell to tissue scale, and then image and segmentation registration of these really rapid image acquisition time resonance scanned images. As far as scientific findings, we found this PCM asymmetry bias towards a thicker PCM below, and this may have a protective mechanism for the cells. And also we found active regulation of the cell volume during osmotic swelling. So for future work, up until this point, everything we've looked at has been studying the solid phase of the articular cartilage. Of course, there's a whole other fluid phase that's very important as well. And we can't see that in our images, but potentially we could, using a technique called Raman spectroscopy, which can measure the ratio between the water and solid content locally in the tissue. And so similar to scanning the fluorescence of the solid phase in the cartilage, we can scan the water content. And we can do that dynamically, potentially with the resonance scanning, and from that essentially calculate fluid motion, which would be quite valuable to articular cartilage mechanics. And then the other aspect, talk about biomechanics, and this has all been mechanics. I want to link it to biology in some way. One way to do that, to see cellular response to, say, something like mechanical loads, is to look at the release of calcium within the cells. Calcium is used to drive most cellular processes. And we do have fluorescent techniques to do that indicate how much calcium is being released into the cytosol within the cell. Historically, we couldn't really link this to the solid phase deformation because we just couldn't measure fast enough. You can measure the calcium flux very rapidly, but we couldn't measure the solid phase deformation on the same time scale. But potentially with this resonance scanning, some future student can spend days and days and years looking into that. And if you could do that, you might be able to look at some sort of measures like time coherence between the biology and the mechanics. Yeah, so acknowledge Walter, Herzog, and Salvatore Federico, my supervisors, Anquan Mu, who's visiting from Europe. He's there. He did the experimentation in his work on the numerical guy. Azim has helped a lot with the development of the software over the years. My supervisory committee, Marcelo Epstein, Steve Boyd, everyone in the Herzog and Federico group, and my funding sources, and the many, many open source software libraries I use in my work. Thank you.
Really nice images too. We will open for the questions. I'll start with you. I'm wondering about the size of the finished
Mhm.
So, does it also depend on where the cells are?
Yes. That's a very excellent question. Very, very, very excellent question, especially from our findings. That fluid motion, the reason it's so high in these models is because we're very close to the draining surface, the top surface of the cartilage. So that's where the water is exiting, right? So the fluid velocities are the highest up there, and also the fluid velocity gradients, that thing that's going to result in the drag of the cell up. So going on our hunch that it's a protective mechanism to have a thicker PCM below the cell and above, intuitively what we expect to see as we go deeper into the tissue is that maybe that asymmetry bias goes away because it's no longer needed because the fluid motion isn't high down there. Of course, the setup that we use, we could only image to about 60 microns deep in these samples. In hindsight, we would have turned the cartilage on its side and image from there, and then we could see cells at all locations throughout. And that is some future work that needs to be done as well. And hopefully, going on the hypothesis of a protective mechanism, we do see a sort of attenuation of that bias as you go with respect to depth.
So mine's a two-parter, right?
Mhm.
At the start, you talked about how there's lots of differences in models for background.
Mhm.
So do you have any thoughts on how you pick the right number? And then on that note, what did you do for your multiscale model to look at the tissue itself?
Okay, so which one do I answer first? I think the second one first. So what I did was just a simple biphasic mixture theory. The permeability, which basically relates how easily water can move through the material, depended on the volumetric strain. So not the simplest biphasic model that you can have, because that is known to be quite important in cartilage. I think that is an important level of sophistication. This wasn't a triphasic model. We didn't consider, say, like the contribution to osmotic pressure of charged molecules in the tissue, or even just solutes at all, solvents and solutes. If we modeled, say, so it entirely depends on the system you're trying to model, right? So if I were to have developed a model of the osmotic swelling experiments, then obviously I need to consider movement of charged particles, because that is what is causing the osmotic swelling there. Well, one of the things. So in that case, you would need a triphasic model. If I'm modeling cartilage in a knee, modeling gait or something, you might not, and I'm interested in, say, the surface, the contact stress, the contact pressures on the surface of the cartilage, I wouldn't even use a biphasic material. I would use an elastic material that is representative at that rate of loading, because that's what you care about. Of course, if you're interested in the cellular deformations resulting from gait and you want a realistic model of the joint, then maybe a biphasic model. There's a balance between models that solve accurately, because also, so this model, it's actually at the higher end of sophistication. It needed 100 elements through its thickness to actually converge, which that's intractable at the level of a knee. That would be 1 billion elements. That would take 1,000 years to solve on a desktop computer. So yeah. They're also biphasic. The cytosol itself, we imagine that the solid phase is basically not there. So the solid fraction was near 0. In that case, it also didn't have any dependence. The permeability didn't just depend on the volume change, because there's no solid to change its pore size, right? The cell membrane, a particularly nice thing about that is that model is there was a new shell element that was released, a multiphasic shell element that can model these sort of material properties in FEBio. And that allowed us to model the very thin membrane of the cell, which historically has been not really feasible, just because of a scale mismatch between that nanometer thick membrane and micron scale cells. So we could model this, like, I think 7 orders of magnitude less permeable membrane. And that's sort of why this is the first time someone observed this translation effect, because you have this impermeable cell that pressurizes, and then you have water moving around it and just dragging it up, right? Because the water can't cross the membrane. And if we didn't have a membrane, we might not have observed that. Yeah.
Ignorant question, Scott. So I had the same question of Venus about the depth dependency of the bottom layer of the PCM, which I think is super cool and makes you want to do experiments where you get cartilage to grow in environments that are completely devoid of mechanical loading.
Mm-hmm.
See what happens. But I thought, couldn't an easy experiment be to still look at the top layer of the articular surface, but look at regions that you think are not heavily loaded regions that are not typically directly underneath
Uh-huh.
the moral condyles, or they're sheltered because of a meniscus or
Yes,
something like that.
that's a very good idea as well. We do know that the difference in material properties of, say, something that's below the meniscus, the cartilage there, can be as much as tenfold versus unloaded regions of the articular cartilage, right? Tenfold stiffer. Going to the point of the paracellular matrix in non-loaded conditions, we actually, in the paper, we take an image from tissue engineering where they were growing chondrocytes and agarose and not mechanically loading them. You get a perfectly symmetrical spherical PCM around the cell. So not in the presence of mechanical loading, symmetric.
Yeah, cool.
Uh,
The other question that I had was dealing with the issue where you do the osmotic swelling, and then you get the cell membrane went back to its original reference form. Maybe it
Yep.
wasn't the cell. Yeah.
the cell itself, yeah.
The cell. So I guess there's I don't know, in my mind, that there could be two reasons why that would happen. One would be it's mechanically protected. You kind of want to get it back to
Yeah.
the reference volume. The other is maybe it's most mechanically sensitive to load when it's in its reference configuration. So it'll respond more to the bulk deformation.
Could be. Yeah.
I was just wondering what your opinion, because obviously the latter would be important for mechanical sensitivity. What you guys are thinking.
Yeah, that's something that actually didn't occur to me. That does make a lot of sense. Does seem to be a regulator, especially the paracellular matrix seems to actively sort of find that Goldilocks zone for cell load cells, say chondrocytes in the superficial zone versus the deep zone. They have very different mechanical environments, but they experience similar strains. And in one of those zones, the paracellular matrix seems to actually amplify the stress of the strain experienced by the cell relative to its local tissue. And in the other, it attenuates it. So it kind of, has opposite effects depending on the depth, for one. That's not the osmotic swelling. That's just like in response to compressive loading. The other thought, and I didn't mention this on the slide, the reason I showed this here is if we look at the surface area to volume ratio, it seems to be flat or maybe even trending towards increasing when the cell is getting more stressed. And going back to like preliminary elementary biology, you learned that the surface area to volume ratio of a cell is very important to its health, right? So if you have a large surface area to volume ratio, you have more of a surface to exchange nutrients and waste across. And so maybe if it's in this sort of harsh condition, the cell might actively try to increase the surface area to volume ratio, which, yeah. But that is another vein too. It's a good idea. Thanks. For someone else to pursue. I'm done.
Your primary goal was to see the connection between microscope
Yes.
and to develop methods that are highly sophisticated.
Mhm.
So in simple terms,
No, I think there's a lot of work to be done still. I mean, it's improved. It's a step forward, but no, I don't think so. And also,
so you will continue to develop the methodology.
yeah, and those hopefully will be applied by other people to do some actual science with them. We did a little bit of science with them with the tools that were developed, but their value would be in, can you do any meaningful scientific investigation with them later? But I wouldn't say that this problem's anywhere close to solved because, well, for one, this whole notion of looking at the cellular scale like we do from a continuum mechanics standpoint is probably wrong because the cell is not a continuum. It has, for one, a cytoskeleton is the main mechanical reinforcement element of the cell. And that's a very discrete sort of system. It's more like a truss cable network as opposed to a continuum. And we know at that scale that things are not in agreement with continuum mechanics. One of the assumptions of continuum mechanics is that we're at a large enough scale, far enough from the scale of like the molecule, that we can make these sort of smooth assumptions and things. So that's one limitation. There's a lot of active biology that we're completely ignoring. We're viewing these cells as these static objects like we do for mechanical engineering. We know that they actively, very rapidly, are remodeling the cytoskeleton on the scale of milliseconds or microseconds, right? So these structures like actin filaments and microtubules are collapsing and then reassembling differently very rapidly. We don't capture that at all. That would be a field of molecular dynamics that we just simply didn't look at. There's many decades of work, maybe centuries, I'd say.
So what are you telling me? Are you telling me that this method is not the right method?
Well, again, it goes back to usefulness and what we're trying to get. This is mostly looking at experimental data, right? So we want to have some sort of measures that we can extract from our microscopic image data mainly, right? And in that case, yeah, I mean, so for example, the asymmetry observation in the paracellular matrix, it's purely statistical observation that was supported by the mechanical modeling, our simplified model using continuum mechanics, and that was able to generate additional hypotheses from that. Is it right? No. But is it good enough to explore something else? Maybe. I don't know. Oh. So of course, the behavior of everything is quantum mechanics on the scale of large numbers, right? But it would be silly to model a table or a chair with quantum mechanics. In fact, you would get huge errors because you'd integrate a bunch of particles and you'd be way off, right? It'd be less useful, more correct, but yes, less useful, right? So I guess that's the art of modeling.
So let me ask another question.
Yeah.
I mean, one of the reasons that people do things like this is to be able to explain the development process, right?
Yeah.
Because if you could answer your question that you indicated at the beginning, that would help to answer.
Yeah.
That question which is not answered at all.
Yeah.
So is there a hope that
absolutely there's definitely a hope.
in which direction do you think is the solution going?
I think this is actually, from a clinical standpoint, too complicated. I think we've actually seen decent success with much simpler models. For example, Don Anderson was here like a year ago. He has a very famous paper where they looked at fractures in the ankle, and those tend to go on to develop osteoarthritis. And they looked at a small cohort, I think about 20 patients, and they used not even finite element modeling. They used what's called discrete element analysis, which is essentially like a layer of springs over the bones that are interfacing at the ankle. And with that simple model, they were able to predict, I think, osteoarthritis in 100% of the patients that got it. And then they were able to predict the grade, the degree of osteoarthritis, the severity, the Orsi grade of it, with something like 90 or 95% accuracy, like amazing, in a very simple model. We're not talking, no poroelasticity, no fluid phase. It was elastic materials, it's springs, and all they were really modeling was the geometry of the two bones. So just from the geometry of the bones, clinical, meaningful, reliable prediction. Of course, cost of that, you can't really make patient-specific models for everyone that gets an ankle fracture yet. So there's sort of economic barriers to that, I think. But yeah, where this might have more value, I think these more complicated sort of studies is in tissue engineering. Not necessarily in clinic, but when we're trying to grow a functional substitute, what sort of loading do you apply to a construct as you're growing it to get the right sort of cell shape, the right sort of paracellular matrix around those cells, the right sort of variation in cartilage properties with position and so on. So, but again, who knows what level of sophistication you need. Mm-hmm. Mm-hmm. Yeah. 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 So an energetically conservative system, we'd expect it to continue to swell. So if you're spending energy, you have an energy source, ATP, in the case of the body, then you might see a difference, which is what we're sort of observing here. Yeah. The asymmetry, I don't think anyone, well, people knew that the PCM in living tissue was asymmetric. They didn't know, at least this bias for the superficial zone cells that we observed, no one had quantified that yet. So no one knew, but I think there probably are ways in tissue engineering that they might be able to exploit that. And in the paper, that's one of the recommendations in the discussion is maybe, so for example, in tissue engineering, there's typically a scaffold that you seed with cells, and the cells grow in the scaffold and replace it with sort of more like collagen and proteoglycans, the sort of molecules that you expect to see in real cartilage. With the scaffold, you have a high degree of control, especially with things like 3D printers and things. Now you can print the scaffold with a very, very refined sort of structure. And you can put drugs in the scaffold. And those drugs, like a growth hormone or something, could influence how much to, like, say, you could kind of tip a bias towards a thicker PCM below the cell, I think, if you, say, seed a little more growth hormone just below the cells and above, for example. Completely independent of mechanics there. And then maybe do some mechanical loading too to help that as well along the way.
The rippling thing is interesting. On the bottom, you use this term strain reservoir. I don't know if I've heard that term before, but can you explain what you mean by that?
Slack.
So it's kind of like maybe it doesn't experience.
It doesn't.
At first, it's just sort of expanding.
Yeah. So if I'm two lipid molecules and I'm in a ruffle, that ruffle's unfolding, the ruffle's unfolding. And then when it's finally unfolded, then I start to, and it turns out you can't go very far before rupture happens. And Kwan has a paper on that goes into a lot more detail. That's an atomic force microscopy I mentioned, yeah. So if we took the very disciplined approach, we would have compared to the immunostaining technique. Of course, we can't do the same image twice, the same volume twice, so we couldn't do a one-to-one comparison. We could do a statistical comparison. We do know that our method likely underestimates the thickness of the PCM by about half. Assuming that underestimation is systematic, the asymmetry ratio should remain the same. And I actually look into that in the paper. If you read in more detail, we scale up, maintaining the same asymmetry in all the models. And that dangerous strain that we think is in the bottom of the cell membrane is actually amplified in the more realistic PCM thickness. So for what it's worth. Yes. So essentially what we're measuring, this stain, we know it binds non-specifically to collagen, just collagen in general. So the majority of your tissue of cartilage is type 2 collagen. In that paracellular matrix, it's type 6 collagen mostly. But the density of that type 6 collagen is much lower than the density of the type 2 collagen elsewhere. And so essentially, we're just measuring the density of the collagen there. And that, of course, is a diffuse boundary. So where do we set that line? I mean, we're consistent, but it could be it likely is underestimating it. Yeah.
That was my underlying question, just the physically that you know is making it so that region is
Yeah, yeah.
Did the speed of loading have any effect on the results?
Speed of loading. So the only dynamic measures we took were those osmotic swelling, which we couldn't really, we could if we had done different osmolarities of the bath. We did the most extreme case of deionized water, so no ions in the water, so it's going to swell up basically the fastest cartilage could swell in those sort of conditions. If we wanted to do rate comparisons, we'd have to vary that osmolarity and that sort of setup. If someone uses a resonance scanning in the future, they could look at other sort of mechanical loading conditions where they vary rate. Chances are it definitely matters. That's my, I shouldn't say it that way. My intuition, strong intuition, is that it matters. Oh, yeah. So for one, at the macro scale, the effective stiffness of your cartilage can vary by 10,000%, depending on rate that you're loading it. You can go from about a 1 megapascal all the way up to like 200 megapascals, Young's modulus. Yeah. So drastic dependence on rate in cartilage. That means for like from a standpoint of health, biology. I mean, different activities might influence your cartilage biology differently. If you're a gymnast, applying 20 Gs of force to your joints, that might be very different than a runner versus a sedentary person that at most walks around. I don't know. We do know that if you unload a surefire way to give an animal osteoarthritis is to unload the joint. Like it will get osteoarthritis. If you put it in cast 6 weeks, it's going to get osteoarthritis. Whereas if you cut its ACL, might get it. Some of the animals will, some of them won't. Some of them won't. Yeah.
But within the context of your work, it almost seems to me like higher loading rate, yes, to cartilage, and of course less fluid exudation through the top layer of the cartilage, which would mean there'd be less of this pull on the lower part of the PCM.
Yep.
It would be almost a good thing from that in that regard.
You're saying at a higher rate, there'd be
at a higher rate of loading, there'd be less potential for that rigid motion of the cell going upward.
Well, when you're that close to the draining surface, we like to say when you load something very fast, the water doesn't have time to move anywhere, but the water's still moving. So when you're really close to the draining surface, you're actually going to have higher fluid velocities there at the higher rate. Yeah, I don't know. There's been work looking at a lot of work looking at rate dependence of cartilage and also chondrocytes, as much as loading rates of like 20,000 hertz. I think Alan Grodzinski at MIT is very interesting. I don't know when you fall off a building or something, load your cartilage at 20,000 hertz, but not good for it to load at that frequency. And, yeah.